Hi, I'm Trevor from the University of Wisconsin-Madison. I'm a graduate student in the Horticulture Department in Wang's Vegetable Sustainability Lab. So right behind me is the NEFA study, and the purpose of this study is to use what's called hyperspectral remote sensing to eventually take data from images, data concerning like wavelengths of light reflected off of the plant canopy, and to relate that data to what's going on in the underground and with the plants. So with the hopes of eventually collecting that airborne data and then using it to predict the ground data. So what we're doing right now with our lab is collecting the ground data ourselves. Our plan originally this year was to have Russet Burbank and Soraya as our two varieties. Right here is our Russet Burbank. You can see this morning we did some digging to get some tuber samples, some of the ground truthing data that we're interested in. And off to the side here we've got Soraya. Soraya is a bit of an issue this year due to some problems that we had around planting. We've got a lot of gaps, a lot of really poor emergence. So we're really focusing on Russet Burbank right now, trying to get as much information as we can out of this. So I guess we can start right here. We have a sensor, we have a data logger with a ton of sensors in the ground. So what's going on with the data logger is a ton of data over time that we wouldn't be collecting ourselves is getting captured here. So data like solar radiation, soil water content, and soil temperature goes here. And along with that, we take data ourselves by hand, and that'll concern leaf area index or emergence, things like that. Throughout the season, we're also taking plant samples. So we're taking samples of the vines, so the above ground biomass. Then we're kind of looking at the leaflets and the petioles, so these leaves coming off of here. And we're looking at the tubers as well. So those are 4 types of in-season data. We're capturing those and then using those to determine the actual ground truth nitrogen status of the plants. So speaking of nitrogen, one part of this study is that we have 4 different nitrogen rates. Right up here up front, we have this rate, we're calling it the control, and it's just the starter. So just the base 40 units of nitrogen that you would apply to the plant. As we go further down the line, we have other nitrogen treatments. Here we can walk through and check it out. You can already see this is the lowest nitrogen treatment, and look at how yellow that canopy is. So canopy color is essential to this project. We're hoping with our hyperspectral cameras, these airborne cameras, we can see changes in canopy color and relate it to things like, in our case, nitrogen status. So we move on, you can see right behind this one is our highest nitrogen treatment. So this is getting 250 units of nitrogen applied throughout the season. Maybe if you can check out both of these, that's a huge difference in the color of the canopy. That's the type of thing that we're trying to pick up with our aerial photos and really correlate how these changes in color specifically relate to the nitrogen status. So like I said, this is the highest, and as we move on, these next 2 treatments, they are the same amount of nitrogen being applied. They both have 200 units of nitrogen, but the timing of this last application is different. So this right here, it received, same as the one before, it received its last nitrogen application just earlier this week, actually, on Friday. So that was more of a late bulking application. But as we move further, this plot's last application was during early bulking. So like I said, between these 2, same amount of nitrogen, 200 units. It's just when that last one is applied. It's the timing that we're concerned with. So this is something that we're interested with our model and with our imaging, not only what the plants look like throughout the season as nitrogen's being applied, but also what changes in how they look if you're applying the same nitrogen in different times. Maybe that affects the canopy color, maybe that's something that we can pick up and prepare for when we start to use our model to make predictions ourselves. So the current way that nitrogen status is measured in a potato crop is, it's not ideal. It's based off of petiole sampling. So it's, people actually go out into the field and they take petioles from the above ground portion of the potato plants, and they take that back and analyze them for the nitrogen status. And the issues with that, for one, it's destructive. You're in the field moving around, breaking things off of plants. It's time consuming because you have to take the plant matter back and analyze it. And finally, it's just one point. You know, you're taking a bunch of different samples and you're seeing what's going on with this plant and that plant, but you're not really seeing the entire image at once. You're trying to more piece it together from different points. So the goal of this project is to use this new cutting edge technology, to use this hyperspectral remote sensing to cut out a lot of these issues. So we hope to eventually be able to take our hyperspectral image and relate that to a model. That model is based off of the data that we're gathering now about ground truthing data. So everything that I've mentioned we're collecting, the model concerns that, and then the imaging taken from above. And eventually we want to be able to use that to just take the image and then predict what's going on the ground. And that way we don't have to worry about all those issues mentioned before. We're not in the field, we're just flying over it. We're not messing with the plants or destroying anything. Ideally, we'll have a much faster turnaround to take our image and see the nitrogen status than you would by analyzing that plant matter. And we'd be seeing the whole field. We wouldn't be getting a few point measurements. We'd be getting a look at what's going on throughout the field, or even throughout an entire farm if a grower was interested in that. So yeah, that's more of what we're trying to accomplish. That's our goals here with the NIFA study.